Moving on to the topic of quantitative trait mapping. So we previously talked about what these quantitative traits are. So they're kind of complex traits controlled by many genes. They tend to be continuous. So think about stuff like height. And we are able to map these through analysis of the genome. So narrow sense heritability, remember we talked about broad sense heritability in the last one, is found by measuring traits over several generations, with a value of 0 indicating no resemblance between the parents and offspring, while a value of 1 implies that the offspring are identical to their parents. Quantitative trait loci are found by comparing the degree of covariation of polymorphic molecular markers with phenotypic trait measurements, which determine genes responsible for the phenotype in a process called QT mapping. Physical maps compare base pair distances along DNA. Genetic maps are based on recombination frequencies, while QTL maps show quantitative trait linkage maps, show linkage of molecular markers to phenotypic traits, and they work best over short distances, and they determine how closely loci contribute to the phenotype. Recombination frequencies for QTL maps are calculated as the percentage of recombination, and we do this by taking the number of recombinants, dividing them by the total number observed, and multiplying that value by 100. A recombination frequency under 20% maps distance directly, but above 20% requires a mapping function. For QTL mapping, a homozygous P generation is crossed, and then the F1 is crossed to give us the F2. We generate a mean for the quantitative trait, and restriction fragment length polymorphisms are used to determine distances between loci. Then they're compared to the phenotype. Moving on to genome-wide association assays, which is our last kind of thing for this section of the topic. So genome-wide association assays are easier than QTL mapping, and they use correlations between single nucleotide polymorphisms, so SNPs, across the genome and phenotype to give a genetic understanding of loci associated with quantitative traits. So we're basically looking for the phenotype of the organism, and we're having a look for which specific SNPs are present in organisms with that phenotype. This gives us an understanding of loci associated with the quantitative traits, but it doesn't exactly tell us the genes responsible for causing the traits, just the SNPs that are usually associated with the traits present. And we typically visualize genome-wide association assays with what I've shown on the right here, something called a Manhattan plot. So that's it for a lot of the annoying stuff for this module. Tomorrow morning I'll just quickly go over the Evo-Devo stuff, talk a bit about entire gene duplication and genome duplication, and then we might finish off on a bit about parental care, and then that'll basically be the end of the module. So that's it. Thank you.